There are a few hands that are up for today, so I'm going to take questions that that some of you have. The first hand that was up is Ajinkya. Ajinkya, please go ahead.
So you stated that the lamina disintegrates to form the nucleoporin, right?
Mm-hmm. No, the lamina is the lamina, right? The nucleoporin is the nuclear pore complex. So that breaks up separately. The lamina obviously breaks up separately. As I said, see, some of this mechanism, like where does individual components go sit, we are still figuring out, right? So the lamina, what happens to the lamina as it breaks up? Does it again get just distribute in the cell and at some point of time reassemble? We don't completely know at this point of time, but lamina and nucleoporins are not the same thing, if that's what.
Yeah. So my question is that you also said that nucleoporins migrate to different parts of the endoplasmic reticulum, right?
Yes.
So is there any significance to that phenomena?
So the nucleoporins are integrated in the membrane. The membrane, nuclear membrane, is connected to the ER. A lot of the distinction between the nuclear membrane and the ER membrane is lost during mitosis, right? So it could still be part of a membrane, the pieces of the nuclear pore complex. Now it could be in a region that otherwise would have been called the ER, right? So a lot of the distinction that exists between many of these structures is kind of altered during cell division and then reacquired as the cell reassembles. So this may just have to do with the fact that the nuclear pore complex is part of the membrane and kind of breaks up and distributes in the membrane and does not fall off entirely from the membrane, right?
Okay, so thank you.
Okay. Vaishnavi, your query.
Sir, in one of the first slides where we looked at how the microtubules arrange during the metaphase, I saw two microtubules from both the centrosomes attached to each other. Why was that?
So this kind of attachment happens, there are many such interactions that happen in the opening up of the two mitotic spindles. Remember, there are strands of microtubules that are connected to them, right? It's not like they are completely, the centrosomes are free floating, because there are microtubules here and there are microtubules here, and they actually are connected. And so along with microtubules that are pulling the centrosomes apart, there are microtubules here that are growing, that are pushing the centrosomes apart as well. So microtubules, as long as they have a way to communicate with each other, and motor proteins are one of the ways they do it, they will have overlaps in places, just as the tip of the microtubules will bind to the centrosome, using a very elaborate mechanism. This kind of overlap attached through maybe motor proteins is also likely to take place, and that's happening because of how this all began, right? So and then once they are sufficiently apart, the microtubules come apart, the chromosomes are arranged, and now that same tips have to go bind the centrosomes, have to bind the centromeres in the chromosomes per se, right?
Right. Okay, sir.
Vignesh, your query.
Sir, so at the start of the cell cycle, you have an approximately spherical cell, and at the end of the cell cycle, you have two daughter cells that have half the volume and that are also approximately spherical. So I did some basic surface area volume calculation, and at the end, you have approximately 25% more surface area than you had at the beginning. So there are two explanations for this. One is that there is a large increase in lipid generation during the cell cycle, the mitosis, that allows the surface area to expand by this much, or the density fluctuates throughout the mitosis that allows for this to happen. So what is happening?
Both probably happen, right? And go look up this review on lipids. I will put it up. And both scenarios actually could be happening. There is a lot more lipid in the cell that now becomes available at the plasma membrane. So, for example, we didn't talk about this, but later on in advanced cell biology courses, you will probably read about it, where there are invaginations in the membrane, like caveolae-like structures, and if you really pull on them, and the membrane has to expand, these caveolae-like structures actually fall back. They give their membrane to the plasma membrane. So there is a way for membrane to be added to the cell membrane, and at the same time, there could be other mechanisms that of synthesis that are also contributing to the amount of lipid that's there in the cells. The two daughter cells may not be as big as the parent cell per se to begin with, and that's something to consider as well when you think about this.
Okay, sir.
Anubhuti, your query?
Yes, sir. I had a doubt in mitochondria breakup.
In what?
Sir,
Mitochondria paper. Okay, huh?
yes, sir. In that slide, it was given that when they break up because of several protein and all, then they rejoin again. So, why do they rejoin?
See, for the functionality of the mitochondria in an active cell, which is not dividing, that architecture is vital, right? So the mitochondria are not existing as elongated structures just for fun, right? So that architecture is vital to how they mediate their functions. So hence, a lot of time and effort and energy is put back in not only, so when distribution and breakup has to happen, those structures are not amenable to that kind of distribution. So you chop them up and then distribute it, and then you bring them back together to being the structure that they originally were, right? Sai Chinmay has a query. How exactly are the organelles distributed among the cells? Is there equal division, or they just statistically get distributed? Stochastically is probably what you meant. Also, are the microtubules involved in gathering all these organelles as well? So a lot of this happens, both of these happens, where some of it is just stochastic distribution. If you have these big, stirring of by the actin, then that obviously ensures that everything is getting mixed nice and well before the division takes place. Some of it is probably also mediated by direct binding to microtubules. And Golgi, for example, there is some speculation that there could be direct binding. So that also could be taking place, right? Anand has a query. What does the term summoylation of DRP1 mean? Summoylation is a modification that happens on DRP1. As a matter of fact, Girish's lab here at ICER studies summoylation of proteins. It's a modification that adds a certain residue, which is called sumo, and hence the name summoylation. That affects the functionality of those proteins that are summoylated, right? So DRP function is affected by summoylation, and that's one of the changes that is taking place. Why is the kind mitochondria being moved around in circles? Because it likes going around in circles, right? So I think the mitochondria are being moved around in circle because it's trying to distribute the mitochondria, right? And make sure that they go they're evenly distributed throughout the cell. What is particularly interesting, and nobody asked me this at least up till now, is why does that movement happen in the anti-clockwise direction and not the clockwise direction, right? Because it moves in one direction. So does it happen the other way too? And if not, why? Right? It happens only in the anti-clockwise direction, so go look up the paper and see what they have to say about why that is the case, right? Abhinav has a query. Does every chromosome always get split properly? Actually, it doesn't, right? Sometimes that actually there can be mistakes, and separation does not happen. Polyploidy kind of situations happen where an additional pair of chromosomes end up in a cell, and the cells don't do fine, and eventually, in most cases, these cells are gotten rid of, but sometimes they can be problematic as well, right? Vinu Kumar, you have a query. I don't know how I should pronounce your first name.
Sir, it's Gadha.
Radha. Okay, go ahead, Radha.
Sir, my doubt was regarding the actin comet tails helping mitochondria. Does that happen only during mitosis, or do they also help in moving the mitochondria during
Yeah, very
normal
good
movement?
question. Very good question. So far, they've been seen only during mitosis, right? And that may have to do in part because of the fact that the architecture of the mitochondria is very different in non-mitotic cells. And so they're not really trying to be moved around, right? And so that might explain why this happens only during mitosis. Many of the mechanisms that required, that trigger this formation of these tails are also activated during mitosis, which is probably one more reason why these mitochondria in the mitotic state only have these tails. They are fragmented mitochondria, and now they are being moved around as well, right? So that's my short answer. Goraksh, your query.
Sir, I didn't get the part where you said it is always anti-clockwise. Like, if we look at it from one side, it is anti-clockwise, but from the other side, it is clockwise, right? Like,
Yeah,
so how do we determine
this is like saying, it's moving in one direction as you look at it. Like, this is like saying, if you put a mirror in front of it, it's clockwise. Sure, when I am looking at it from, you're looking at it on the screen, it moves in one direction, right? And it doesn't matter what that direction is. You can call that direction, whatever you want. But there is movement in of the actin ring in one particular direction and not the other. And that's the query. What on earth lets the cell say, this is the direction I'm going to move this in, right? And clockwise, anti-clockwise is our terminology. It doesn't really matter. Okay. Couple of questions on chat. Dhairya has a query: why does the cell try to go into a spherical structure before mitosis? Good question. What do you think might be the reason? So is that when it comes to separation of two daughter cells, having a rounded cell ensures that a lot of the heterogeneity that exists. See, when the cell is spread out, deciding where the central plane for division is may not be that easy to do, right? That's a lot more easier to do if the cell is rounded up, right? Because you kind of have a sense of where exactly that middle is going to be like. This may also, obviously, the finding of that middle is determined by lipids, for example, that could be present, and their distribution is again regulated differently when the cell is in this rounded conformation. So to allow for this kind of separation to take place and ensure as close as possible for even distribution of components, because many of these components have some active mechanisms, but for the most part are using stochastic methods to be distributed. So you want that separation to ensure that there is a 50-50 chance of things going this way or that way, and that in part mediated by or facilitated by the fact that cells round up. So there is a active mechanism to reduce the adhesion of the cells and allow them to round up before this happens. Okay.
Thank you.
Sorry, you were saying something.
No, I was just saying thank you.
Okay. Gautam has a query: are mid bodies formed in telophase permanent? If they aren't, then how do the microtubules and actin fibers within them get distributed? So there is a mechanism for everything, right? And as I said, I have not gone into the detail of many of these aspects simply because the scope of that is just too much. How are the centrosomes duplicated? Again, very detailed mechanism is known about how this is taking place. So there are mechanisms for these structures to break up, just as there are mechanisms for them to be brought together and assembled into very distinct architectures. And that mechanism, when it comes to the Golgi or the we briefly touched upon how phosphorylation, dephosphorylation of very specific proteins could do this. So if you're curious, go back and read a little bit. There is a lot of information on how many of these processes are regulated as well. If the microtubules, Nilanshi has a query, if the microtubules attached to the kinetochore after the chromosomes are arranged at the metaphase plate, how are the chromosomes arranged in the first place? Please go look this up, okay? How is that architecture, created? I'm not discussing that here because that's really beyond the preview of this class. There is a lot of information on how this happens. Right, each and every step. I could teach an entire course on just cell cycle, right, and all the steps. So go look this up and let's see what you find out. Right, last two questions. Hirak, you go first.
Sir, in continuation to Gorakh's point that if mitochondria move in anti-clockwise sense during cell cycle, but the cell could turn upside down and then the mitochondria would be moving in the other.
It doesn't matter. This is what I'm trying to tell you. It moves in one direction, this way. Now, if you do the cell this way, it is still doing the same direction, right? It's relative to the cell, its position may have changed, but it moves in one direction. That's the point, right? So you can look at it this way, you can look at it this way, and clockwise, anti-clockwise is our view of that, right? But effectively within the cell, that movement happens in a certain direction, right? And it's not like it goes one round this way and then goes another round that way, or it does 5 rounds this way and then goes back the other way. It does rounds in a certain direction and keeps doing rounds in that direction. And the question was, why that could be happening at all. Okay, go look this up. They don't have a clear explanation for this, but they have some suggestions on what could be the case. Vaishnavi, last query.
So during the actin wave, does it move around only the mitochondria or other organs also?
So the mitochondria have active tail-like structures that allow it to be moved. But the pieces, the fact that you can see this big wave of actin that is going through suggests that other structures, which are pieces that are floating around, will obviously be moved around because there is a cytosolic current that is created effectively, right? And so that will ensure that a lot of the other broken up structures are also distributed in the cell, right? So this could have implications for other structures in the cell and the way they are distributed also. Okay, so yeah, so mitochondria could be the place where this was discovered, but it probably has implications for other cellular components being distributed as well. Okay.